more delighted to welcome him in our family here. And hopefully the future is bright. But with more introduction, we're going to Susie.
Hey, sorry. Hey, good morning, everyone. It is a great pleasure to introduce today's speaker, Dr. Albert Spada. Dr. Spada is a leading physician scientist in the field of neurodegenerative disease research. He received his MD PhD training at the University of Pennsylvania. And while he was an MD PhD student, he identified the cause of X-linked spinal and verbal muscular atrophy as an expansion of a trinucleotide repeat. And after he completed his residency and postdoctoral training, he became a professor at the University of Washington in 1998. And in 2009, he became a professor and division head of genetics in pediatrics, cellular and molecular medicine, and neurosciences at UCSD. And in 2017, Dr. Spada was selected as the founding director of the Duke Center for Neurodegeneration and Neurotherapeutics, and also as the vice chair for the research in the Department of Neurology. And in 2020, he joined UCI as a distinguished professor in the Department of Neurology and as the founding director of UCI Institute for Neurotherapeutics. Dr. Spada and his lab have made numerous key findings that are important in understanding the pathogenesis of a number of neurodegenerative disease, which have been published in a number of high-profile journals such as Nature Cell Science. Dr. Spada has been the recipient of many prestigious grants and awards from NIH, HHMI, American Society for Human Genetics, Harrington Discovery Institute, and he was also selected for the Chan Zuckerberg Initiative Neurodegeneration Challenge Network. Today, we are very honored to have him this morning, so please welcome Dr. Spada.
Okay, so first of all, can you guys hear me? I guess that's the most important thing.
Yes.
Yes, very well.
Awesome. And let me go to slideshow. Okay, and let's see, does that advance okay?
Yes.
Yep. Great. Okay, so well, thank you, Susie, for that very nice introduction. And Chris, it's a pleasure to be speaking in this seminar series, which I think has been incredibly successful and exciting. You've done a wonderful job, as well as the organizers working with you to put together really a wonderful respite from, the insanity of the pandemic every Friday morning. So yeah, now today I thought what I would do is tell you about our research on Spinocerebellar Ataxia type 7. As you'll learn, this is a disorder that has a very interesting form of retinal degeneration associated with it. And I'll cover our work on the retinal degeneration as well as some of the work on the cerebellar degeneration to give you the whole sort of picture of our research on this particular disorder. And thank you all for attending. And of course, I look forward to your questions and comments at the end. Okay, so as Susie mentioned, a long time ago at a medical center far away, I was an MD PhD student, and I was fortunate enough to connect with Kurt Fischbeck, who's a branch director at the NINDS now, and I became interested in this disorder known as spinal and bulbar muscular atrophy, or SBMA for short. And the goal of my project was to identify the causal gene. And I was fortunate. I found that there was a causal mutation in the androgen receptor gene. However, what was really exciting is we discovered that this disease is caused by the expansion of a CAG repeat, a variable repeat that occurs in the first coding exon of the androgen receptor gene. And this was the very first repeat expansion mutation to be identified in any human disease. It really preceded the discovery of the Fragile X mutation, though their paper was published before ours for reasons that I could explain to you at the end if anyone's interested. But anyway, be that as it may, what we discovered is there is the CAG repeat that roughly doubles in size, and CAG encodes the amino acid glutamine. So this is a disease that involves a polyglutamine expansion in the androgen receptor protein. And this was the first CAG-polyglutamine disease to be described. And now we know that SBMA belongs to a category of at least 9 disorders that involve polyglutamine expansions. The best known, of course, is Huntington's disease. And in terms of the greater repeat expansion disease category, there's roughly 40 diseases that are caused by all sorts of different repeat expansions, mostly trinucleotides, but you also see tetranucleotides, pentanucleotides, and hexanucleotides causing disease. Okay, so in terms of CAG polyglutamine neurodegeneration, I think that we were able to advance rather quickly in our understanding of the pathogenesis of these disorders. So of course you have too many repeats at the DNA level. You make an RNA that has too many repeats, but you end up making a protein that has too many glutamines. And this really is the crux of the pathology. Once you have a length of glutamine tract that exceeds about 35, what happens is that the polyglutamine tract misfolds. It adopts an abnormal conformation. And this is the initiating step in the pathogenic cascade. And in fact, we can generate antibodies that detect this misfolded conformer, and those antibodies will cross-react with all the different polyglutamine disease proteins. And so what we think is going on is that disease is caused by a toxic property or properties that are associated with the misfolded polyglutamine expanded protein rather than simply a loss of protein function. And as many of you know, when you have these misfolded conformers that are soluble, they can then become oligomers and then ultimately become insoluble fibrils. And sometimes you see inclusions or aggregates at the light microscope level. Now, the work we did on this and related rare disorders in the mid-90s coalesced with work that was going on in Parkinson's disease. And a really important realization occurred. And this, I'd say, was a paradigm shift in our understanding of neurodegeneration. The field came to realize that pretty much all neurodegenerative disorders, whether they be exceedingly rare disorders like the prion diseases, or very common disorders like Alzheimer's or Parkinson's, that they always involve proteins that misfold. Now, of course, it's different proteins in the different diseases. And then what happens is the misfolded proteins accumulate and form aggregates and inclusions. And in the different disorders, those aggregates and inclusions, accumulate in different subcellular localizations, different regions of the brain, different regions of the spinal cord. And that pathology then has been described, and it's been sometimes named by famous pathologists, or, so we have all sorts of histopathology related to aggregation that's been described. And so this was a really important realization because it also has therapeutic implications. What it tells us is that in our central nervous systems, we are predisposed to having a problem with protein quality control. And so if we can come up with a therapeutic intervention that would somehow rectify this impaired protein quality control, we might have a therapeutic approach that would treat all of these diseases. However, there is another aspect of the pathogenesis of neurodegeneration that's been really a conundrum for our field. And it still remains, I'd say, a key question in neurogenetics research, and that is, what is the basis of selective vulnerability? So by selective vulnerability, what I mean is that if you consider different disorders, which are either strictly inherited or can be inherited, what you see is that different regions of the central nervous system, are affected in the different diseases. Yet, in each of these cases, you have a mutant gene that leads to the production of a mutant protein that's expressed throughout the central nervous system. Indeed, in the case of ALS1, that's caused by mutations in superoxide dismutase 1, every cell in the body sees the misfolded protein, yet only motor neurons in the brain and in the spinal cord degenerate. Now, this is a really important problem. And so when I was a postdoc, I thought, oh, well, isn't it interesting that there's this disorder that has retinal degeneration, and maybe we could understand selective vulnerability if we focused on SCA7 retinal degeneration. And in fact, that was the reason why I decided, to work on SCA7 in the first place. Okay, so here I am swimming off the coast of Baja, California in happier times. And, we're in a tough business, so you need to learn how to swim with the sharks. But if you're going to swim with sharks, I recommend you swim with the whale sharks because they're filter feeders, so you have nothing to fear. And, we're pretty close here, so you can hopefully in the future go down to Baja, California and do this yourself one day if you're interested in so doing. Okay, so Spinocerebellar Ataxia type 7 is an autosomal dominant cerebellar ataxia, and it's readily recognized clinically because many patients also show retinal degeneration. There's also another prominent genetic feature known as anticipation that I'll explain to you what that is. The gene was mapped to chromosome 3p back in 1995. It was cloned in 1997 as a novel gene, so it was given the name ataxin-7. It encodes an 892 amino acid protein of what was at that time unknown function. And you see the stereotypical domain architecture here for ataxin-7. The polyglutamine track begins at amino acid 30. There are these conserved blocks that you can actually trace the conservation back to yeast. And it actually traffics between the nucleus and the cytosol. And so it has functional nuclear localization signals and nuclear export signal. Now the CAG repeat range at the ataxin-7 locus is from 7 to 34 in individuals who are normal. And the disease range starts at 37 and goes up into the 400s. You can even have cases of infantile onset SCA7, which I've been involved in actually 3 of them this past year, and they're very upsetting and tragic, needless to say. So the pathological hallmarks of SCA7 relate to the cerebellar degeneration, as you can see here on this MRI, and then there's this retinal degeneration. And when you look at these images, it looks like a macular degeneration. But in reality, it's a cone rod dystrophy. And the patients present with problems with color vision and then central vision, and then they go blind. There's a lot of variability. In a paper that we published recently, we sort of discussed this. And you can see here we have a patient 55 years old who has 40 CAGs. So this is a natural history study that we started like 5 years ago at the NIH, at the NEI. And we've been following these patients now for 4 years. And so this is, you can see from the OCT and from the microperimetry that this individual is only mildly affected. This patient presented with some moderate disease, as you can appreciate here. And then over the course of 3 years, you can see how much this disease advanced. And you can see serious problems in terms of the performance on the microperimetry test. So you see this variability in terms of disease severity, and then you see this progression that you can monitor over a relatively short period of time, unfortunately, in many of our patients. Now, when I was a fellow at the University of Washington in clinical genetics, I had the good fortune of working with Tom Bird, who's an outstanding neurogeneticist. And I was involved in the care of this family with SCA7. And what you see here is that the disease is getting worse as you go from parent to offspring. That is what we call anticipation. So when the disease gets worse in terms of age of onset and rapidity of progression, we call that anticipation. And anticipation historically is rather interesting. It was first described by a German physician caring for patients with myotonic dystrophy over 110 years ago. And there was some question as to whether it was real in the middle part of the last century. But then when these dynamic gene mutations were identified, it was clear it was real, and then we had a genetic explanation for it. So in this case, this individual here, I assume you can see my arrow, who died at 63, he presented at around 50 years of age. He had a tremor, and he saw a neurologist, and the neurologist was like, oh, stop drinking so much coffee or whatever. Didn't take a family history, of course. The patient got worse, and it was a real problem for him because he was a cardiothoracic surgeon. So another neurologist saw him, took a family history, and learned that he had had a child who had had ataxia and had died at age 16, and said, oh, I think you may have one of these inherited ataxia syndrome. So that's how he came to our clinic, and his disease progressed, and he died at 63, and his other two children presented in their 20s. And then we followed them, and they both subsequently passed away. And his mom, who at the time he was being evaluated, it was clear she had had some problems with coordination, but they were very mild in that she lived a normal lifespan. So what you see here is onset in the 60s, onset at 50, and onset at 6 or in the 20s. So that's very extreme anticipation. And that is something you see in SCA7. And you can imagine it's very challenging in terms of caring for these families because it's a very tragic disorder for many of our families. Okay, so in terms of the photoreceptor degeneration, as I said, patients, the problem initially is really with loss of color vision. The very first sign is you can't distinguish yellow from blue. Central acuity goes then next. If you do ERGs, you see marked dysfunction of cones in the presence of normal rod function at the initial stages of disease. So that's important in terms of differentiating it from macular degeneration. The rods do, however, also eventually degenerate, and then patients lose both central and peripheral vision, and they go blind. So SCA7 is a bona fide cone rod dystrophy. It's not a macular degeneration, and in fact, the last time I reviewed this, there were only 9 genes that had been identified as causing cone rod dystrophy, and ataxin 7 polyglutamine expansion is one genetic way to get there. So when I started my lab at the University of Washington way back when and became interested in SCA7, I said, okay, well, we should generate an animal model to try and understand the molecular pathology. And so it was like, okay, what model organism should we choose? Well, you got to be careful. Professor Zerkowitz went with the crossbred flying fish and piranhas, and that was really a mistake. I love The Far Side, by the way, for those of you who remember it still. So we went with something much more traditional with the mouse. And our approach was a standard transgenic approach. We used this murine prion protein expression system developed by David Borchelt. We knew that it would drive expression in the neural retina, and that was important for our work. And we developed 2 constructs, one with a normal length repeat and one with an expanded repeat, and we generated lines of transgenic mice. And we characterized those mice, and it became clear when we did retinal sections, and this is using Richardson stain, that something was awry because you could see this sort of, of course, this beautiful stereo architecture here, cytoarchitecture, I should say, of the retina that I still love looking at these images, never tire of that. And you can see this scalloping where, the outer nuclear layer, the nuclei, the photoreceptors are scalloping, and the number across is diminishing here in this periodic pattern. So there's a dropout there. So we wanted to see, okay, well, which photoreceptors are going? And so we did retinal whole mounts, and we used an antibody against cone photoreceptor, and you can see that the 92Q animals are bereft of cones. And this is just sections where we use confocal imaging, and our cones here that were against a cone photoreceptor pigment. You can see we can just detect 2 lonely cones here in this section compared to, in our controls. So this suggests that histopathologically cones are dropping out. But in terms of visual function, is that true? So we did, with the help of Jim Hurley, we did electroretinograms. And what I want to emphasize is initially you can see that function is normal, but you have a degradation of cone responses ahead of rod responses. And this is exactly what you see in human patients. Ultimately, of course, the mice go blind, and you don't have a signal from cones or rods. But this histopathology and electrophysiology really closely recapitulated what you see in human patients, which we were very pleased about. So we decided, okay, let's see if we can now use this model to understand the molecular basis of the retinal degeneration. And so we thought about how to approach this. And again, I thought about the genetics, other genes that are involved in causing cone rod dystrophy. And I came across CRX, cone rod homeobox protein. And this was cloned by Shiming Chen using a yeast 1 hybrid screen with the rhodopsin promoter element as bait. It's a 299 amino acid protein, has a molecular mass of 39 kilodaltons. And importantly, it has a polyglutamine rich domain. The reason why that's important is that polyglutamine domains in proteins and transcription factors are points of physical interaction. And so, I hypothesized that perhaps CRX and Ataxin-7 were physically interacting. And perhaps that was the basis for the degenerative retinal phenotype. So CRX binds to a particular consensus sequence in the promoters of various photoreceptor expressed genes. And importantly, mutations in the CRX gene can cause an autosomal dominant form, cone rod dystrophy type 2, autosomal recessive loss of function mutations cause Leber's congenital amaurosis. And you can evaluate CRX transcription function readily using a rhodopsin promoter reporter construct. So we tested the hypothesis for physical interaction. This was a collaboration with Shiming Chen at Washington University. And here we just did a yeast 2 hybrid, a direct one-on-one sort of set of experiments. And we saw evidence for a productive interaction for Ataxin-7 with CRX, as you can see here. And we did in vitro co-immunoprecipitation studies as follow-up to confirm, and we were able to confirm the physical interaction as indeed being direct. So then we looked at our transgenic mice. And back in 20 years ago, it was like a big deal to do real-time RT-PCR. It seems sort of quaint now, in the days of next generation sequencing, of course. I guess I'm aging myself here. But anyway, what we did is we studied, we took RNAs from retinas from mice who were pre-symptomatic for disease. And we basically selected a number of different photoreceptor expressed genes, including ones that were CRX targets and ones that were not. And all the CRX targets showed significant reductions in the expression, whereas the non-CRX targets did not. So we hypothesized that SCA7 retinal degeneration is resulting from interference with the function of CRX in the retina. And so we were interested in really understanding more about Ataxin-7 normal function. And so just to remind people that transcription is a very carefully coordinated process. The transcription activation complex often involves over 150 proteins coming together. And the proteins, of course, come in different varieties. And the green ovals in this diagram would represent specific transcription factors, that bind to sort of DNA recognition elements. Now, it's known that it's important for them to interact with so-called co-activator complexes that have chromatin remodeling activity. And one common chromatin remodeling activity is histone acetyltransferase activity, or so-called HAT activity. So there are a number of proteins that have HAT activity, and they're listed here. And so GCN5 is one of them. And so in terms of co-activator complexes, there's a co-activator complex that includes GCN5. And GCN5 was originally identified in yeast. And this complex identified in yeast was found to have HAT activity for histones, but only if GCN5 was there as part of the complex, and histones and nucleosomes. So not GCN5 in isolation. And this complex is known as the so-called SAGA complex. Now, as it turns out, these individual components identified in yeast are conserved in humans, and they were subsequently cloned. And in humans, we have a cognate complex that is distinct from the transcription factor 2D coactivator complex, which is, again, one of these major coactivator complexes in mammals. And this so-called co-activator complex is known as the Spada complex. So as it turns out, the ataxin 7 homolog is SGF73. And if you do pull downs, if you do immunoprecipitations in yeast, and you basically pull down, SGF73, you're able to identify the various components of the Spada complex. So, we wondered if, therefore, ataxin 7, in mammals, would be part of the Spada complex. And, these are very complicated biochemical questions. So I turned to Bob Rader at the Rockefeller University for help here. And he was interested in the Spada complex. And then looking over his sort of biochemical purifications and mass spec analysis, there indeed was a protein of 110 kilodaltons that turned out to be ataxin 7 as part of the Spada complex. And this was confirmed. If you used our antibody against ataxin 7, you can pull down components of the Spada complex. And we looked at this further, and what we found is that you can even, pull down the histone modifying activity. So here what you see is if you IP ataxin 7 from hex cells, and then you look to see if you can acetylate histones, H3 and H2B, are acetylated readily, as if you would with something a core member of the Spada complex. So then we went on with the Rader lab to generate stable cell lines, hex cells that express either normal ataxin 7 or the polyglutamine expanded, and we repeated this histone acetylation assay, and we saw, sure enough, if you have normal ataxin 7, no problem in terms of doing histone acetylation. But if you express the mutant protein, you have an inhibition of the acetyltransferase activity. And then we went to mice, and we said, okay, well, let's see what's going on in terms of histone acetylation and GCN5 occupancy, promoters of photoreceptor expressed genes. And if you look in the liver, of course, you don't see any evidence of occupancy or of the acetylation mark. But if you look in the retina, you see occupancy and you see the mark. In the proper regions, if you look in regions where there's no regulatory activity, you don't see any evidence of occupancy or acetylation. And if you look in SCA7 mice, you see in the pre-symptomatic mice that there's occupancy by CRX, and there is the acetylation mark in the promoters of many photoreceptor expressed genes. And I've highlighted a few of the ones here where you see this most prominently, whereas the disease progresses, you have a reduction in the histone acetylation, and this correlates with reduced expression. So this suggests that SCA7 transcriptionopathy may involve interference with CRX activity because CRX is a Stada-dependent transcription factor. So just to summarize what I've told you in this first part of the talk, ataxin 7 is a transcription factor. It's a core component of the Stada co-activator complex. Polyglutamine expanded ataxin 7 gets incorporated into the Stada complex and inhibits GCN5 histone acetylation. We showed that all three of these proteins directly interact with one another. I showed you the chip analysis where you see that ataxin 7 and GCN5 occupy CRX gene regulatory regions. You see how histone H3 acetylation status correlates with the occupancy. And also that if you look at SCA7 transgenic mice, there appears to be this problem with the histone acetyltransferase activity of Stada. So based upon this work, we proposed this model. Ataxin 7 is part of this co-activator complex, and it incorporates into it. And there are certain transcription factors that rely on Stada for activating their target genes, and CRX is one of them. There's probably a number of others, and that's something that we're interested in. However, when you have a polyglutamine expanded ataxin 7, it goes into the complex. It does so in stoichiometrically appropriate ratios, as we showed with the Rader lab. However, what happens is then you have an inhibition of chromatin remodeling activities, including histone acetyltransferase activity. So transcription factors that rely on SCA co-activator activity can no longer activate the transcription of their target genes. Okay, so we're going to shift gears now. So this was...
excuse me. So can I ask a question regarding this first part?
Sure. I'm an easy...
Maybe you will continue, so maybe you will answer my question, but did you observe inclusions of the GCN5 in the nucleus, like basically blobs of GCN5 in the nucleus? And well, basically today we would call it phase separation, right? Because it seems like this is what those those repeats are doing on the protein level, that there's repeats of the one amino acid. It's unfolded and unstructured elements. So today I think we would call it phase separation, right?
Well, first, yeah, so if you look at the aggregates in the nucleus, there's many different proteins that are there. Components of, protein quality control pathway, a lot of ubiquitin subunits and things like that. Heat shock proteins, etc. And GCN5 is there also, for sure. Yeah, now in terms of, so polyglutamine expansions, they're prion-like, but they're not true prions is what we think. And so, yeah, it's weird. They do have a certain domain architecture. They're not folded. So, yeah, we don't really think of them in terms of phase separation as we would for other sort of entities that have those low complexity domains. So it's a different type of biophysics, if you will, that appears to overlap in some way with the phase separation, but is distinctly different. You wouldn't see that like sort of based upon temperature that they would go in and out of solution like you see classically. With the liquid, liquid.
Yeah. It's,
did you look on the structure by right now cryo-EM, let's say?
No, we haven't done that. So, yeah, it's been very hard actually to do any sort of crystallography on
Not crystallography, but EM.
Cryo-EM. So cryo-EM, we can certainly use that, and but we have not done that, and I think that would be interesting. These questions actually remind me of how I went to the very first Gordon conference on, polyglutamine disease. And I had the pleasure of meeting and getting to know Max Perutz. He was like in his mid-80s at that point. And he became very interested, and proposed this polar zipper concept that you get this beta sheet that folds back on itself with the polyglutamine tracks. And he published a few papers about this. And this still is what people feel at a structural level is occurring. Yeah, I haven't heard of any cryo-EM work, nor have I been involved in it, but I would love to discuss, such work with you further, Chris. That would be great.
If I may come back to the nuclear function of ataxin 7, since it is in and out, it's probably regulated by some signals. But one of the things that came to my mind, it will obviously change the organization of the nucleus if we start to have those, especially lack of acetylation of histones. I mean, it obviously will. Have you ever thought, or maybe somebody else looked at the organization of the nucleus, especially with the progression of the disease or progression of number of repeats?
Yeah, there was a very nice paper from a French group in ELS biology where they showed that if you look at the retina, you see a decondensation. You could see it at the light microscope level, so that the nuclear chromatin, starts to, decondense. So it's almost, it's interesting because there's two sides to this coin. It appears that on the one hand, you have a suppression of gene expression for certain genes, but then you have other genes because of this improper chromatin remodeling. And there's another chromatin remodeling activity that I'm not talking about here, histone H2B deubiquitination, which involves USP22. that's in the complex also. And it appears there are other genes that get inappropriately expressed because you have a loss of heterochromatin in certain regions of different chromosomes. So yes, so there's a lot going on there.
Okay, thank you.
Okay, so back to my friend Philip the bear. Again, I do recommend after the pandemic, if you want to go camping, going to Sequoia National Park. It's really beautiful. But if you visit and you leave food in your car, and overnight, like Philip the bear smashes your windshield to take the food out of your car, the park ranger doesn't get angry at Philip the bear, just thinks that you're a moron. That didn't happen to me personally, but someone else in the party. So, but Philip actually looks intimidating, but he really isn't. But again, a really wonderful part of California to visit at some point. Okay, so just want to tell you a little bit more about our work on SCA7 beyond the retina, and then I'm going to return to the retina and talk about therapy development for SCA7 retinal degeneration. So my former graduate student, Jackie Ward, led this study, and it occurred to us that patients with SCA7 have a number of disease phenotypes that you would see in bona fide mitochondrial diseases. We did this mitodb.com algorithm, and the unbiased hierarchical clustering showed that SCA7 clinical features would give a mito score of 93 out of a score of 1 to 100, which is very high. And it's interesting when you do the clustering, it sits here, around a few diseases, including Leber's. We just very briefly, it's clear that there's some sort of metabolic defect in patients of SCA7. In this task, this is a visual task, and when you're doing magnetic resonance spectroscopy using P31, you can monitor the production of inorganic phosphate as a ratio of phosphocreatine, and controls, based upon that brain activity, you see an increase in inorganic phosphate, but SCA7 patients, you see no such increase, suggesting that there's a problem of a true metabolic nature going on in the brain. And what we did in this study is, when I was at UCSD, I basically became interested in using stem cells to model human disease, and we've done this for SCA7, where it was generated from fibroblasts. We reprogrammed them to iPSCs. Everyone knows this technology, and we made neural progenitor cells. What we did is we were interested in looking at nicotinamide adenine dinucleotide, or NAD. And so I contacted Lulu Cambron, who was at the Volum, and she developed this very nice ratiometric biosensor for NAD, where when it binds NAD, then the fluorescence at 488 is extinguished. However, there's excitation of the complex just by virtue of its existence at 405. So you basically can quantify NAD very carefully in vivo by seeing a reduction in the fluorescence of 488. As you can see here, a reduction of fluorescence correlates with increased concentration of NAD. And so what we did, and then this is linked to localization signals, so you can direct it to the nucleus, the cytosol, and the mitochondria. So we did that in the MPCs from patients and controls. And what we found is that the ratio went up significantly in the nucleus and the mitochondria, suggesting that NAD levels are reduced in neurons derived from SCA7 patient stem cells. So that was something that we published, and this will dovetail with work that I'm going to tell you about now also, and that is work from my former graduate student, Colleen Stoyas. So hopefully I convinced you that SCA7 is a transcriptionopathy. So to understand the cerebellar degeneration, we decided we would do unbiased transcriptome analysis, and we used SCA7 transgenic mice that we had generated, actually a different set than the ones I presented earlier. And we basically isolated RNAs from the cerebellum, both from pre-symptomatic and from symptomatic mice. And we identified about 100 genes whose expression was significantly altered. And what was very interesting is that we had a clear, I mean, you didn't even need to run a bioinformatics algorithm to see that you had this incredible enrichment in a coordinate downregulation of genes that are important for regulating calcium homeostasis. Now, when you think about calcium in the cerebellum, when you think about the proteins in the cell membrane, and the cytosol, and the endoplasmic reticulum that regulate calcium homeostasis, it's clear that this must be important for cerebellar function, because mutations in many of these genes cause ataxia, either in humans or in mice or in both. So this would seem to be something that would be important for understanding SCA7 degeneration, we thought. Now, we wanted to go further in terms of the mechanism. So we took those calcium regulatory genes whose expression was altered, and we ran this opossum algorithm. So with this algorithm, you're basically looking at the promoter sequences of the downregulated genes, and you're looking for shared transcription factor binding sites that are enriched. And we ran that analysis, and we identified a number of transcription factor binding sites. These are our top 11 hits. And among the top 10 hits, what stuck out to us immediately was like the PPRE, the PPAR response element, and the hypoxia response element. And the reason why that caught our attention is that the transcription factors that bind to these sequences are regulated by sirtuin-1 or SIRT1. And the reason why sirtuin-1 is so interesting in neurodegeneration, and by the way, sirtuin-1, for those of you who don't know, is an NAD-dependent deacetylase that links metabolism to longevity and neuroprotection. And about 20 years ago, it was shown that SIRT1 orthologs in yeast could extend lifespan, and that was extended to worms and flies and mice. There's some controversy around this, but I think that the data still holds. But what's not controversial is that SIRT1 overexpression is neuroprotective. So you see that in models of Alzheimer's, Huntington's disease, and SCA3. And there's been a lot of discussion of how that neuroprotection works, and lots of ideas in terms of target genes that SIRT1 is activating. But in reality, it's not well understood how it works. So we wondered if this sort of calcium connection could be relevant to SIRT1 neuroprotection. Well, we went to our SCA7 mice and we looked in their cerebella, and what we found was that one SIRT1 target, PGC1 alpha, we looked at its acetylation status, and we saw that there was markedly increased acetylation in the cerebella of our SCA7 mice, suggesting decreased SIRT1 enzymatic activity. Getting back to NAD, so it turns out that SIRT1 is NAD dependent, and PARP is also, so poly-ADP ribosphosphate is NAD dependent, and they compete for NAD in the nucleus. And so we wondered if somehow there could be a problem in terms of NAD being reduced because it was SIRT1 was being outcompeted by PARP. So that's something we considered, and we saw evidence for increased PARP1 expression in the cerebella of our SCA7 mice. And we did very detailed measurements, actually, using mass spectrometry. And we could see that there was a trend toward NAD reduction in the cortex, but a significant reduction in cerebellum. And we also took cortical neurons from our SCA7 mice, and we saw that if you look for evidence of DNA damage based upon gamma H2AX staining, that it was increased at baseline, but if you sort of stressed with hydrogen peroxide, that increase became much more dramatic in terms of the DNA damage. So we thought, okay, let's sort of test some elements of this sort of model. And one element is if SIRT1 function is diminished, can we rescue SCA7 disease phenotypes in the cerebellum by overexpressing SIRT1? So we obtained these mice that David Sinclair had generated that are flock stop SIRT1, and we basically made mice, we crossed them with CMV Cree, and we made mice that expressed SIRT1 ubiquitously, and we showed importantly in the regions of interest for SCA7 that we got 3 to 5-fold overexpression of SIRT1. And then my poor graduate student had an indoor, not just one genetic cross, but 2, where basically we took the SIRT1 overexpressors And we crossed them with the 92Q mice, the floxed ones that I showed you for the transcriptome analysis that have actually slowly progressive disease. And then the 266 knock-in model that we have that is a rapid progressive line. And when you do these crosses, you generate 4 types of progeny in a 1-to-1 to 1-to-1 ratio. And we're interested in comparing the SCA7 mice to the biogenic mice. And of course, we have the controls as a basis of comparison as well. And what we observed is that overexpression of SIRT1 could rescue cerebellar degeneration. As you can see, these are sections of the cerebellum, which also has beautiful cytoarchitecture. This is the so-called molecular layer. This is the Purkinje cell layer, where you see the Purkinje cell soma. And then these are the granular cell nuclei of the granular cell neurons, which actually are the largest neuronal component of the brain, as it turns out. So what you can see here is that the dendrites of the Purkinje cells are degenerating. You don't see as much calbindin, and also the soma size is going down. And we could rescue those phenotypes by virtue of SIRT1 overexpression. And in this very aggressive model, we could extend lifespan quite significantly. It was about 11 days, but that's quite significant because the mice only live about 11 weeks. And then in the model where the progression is much more slow, if we have this, this neurology functional sort of exam that we've published in Jove. Sadly, it's one of my most highly cited papers. But anyway, be that as it may. So we use this paradigm to analyze the mice, and we could see that there was an improvement in phenotypes of the mice that were because as the score goes up, you're getting worse. So you see an improvement of motor phenotypes. And then what we wanted to do is we wanted to go back to those genes that we identified whose expression was altered, and we decided we would use the nanostring platform. It's a digital array, and we arrayed the 100 genes on a custom chip, and we looked to see if their expression was altered. And so we went back to those 100, and we validated 96 of the 100 as having the expression changes that we predicted. And then we took the biogenic mice that overexpressed SIRT1, compared them to the SCA7 mice, and we found that 58 of those 96 genes that their expression was rescued, which is quite significant. So the transcriptionopathy was improved as well as the pathology and the phenotype. Well, the other aspect of the phenotype that I didn't show you is that there's electrophysiological abnormalities. If you prepare acute cerebellar slices from SCA7 mice, and you measure the spiking regularity, you see it's quite irregular. And that's rescued nicely in the mice that overexpress SIRT1. And here what we're looking at is the after hyperpolarization that follows Purkinje cell spiking. And that is, the decay of that is significantly more rapid in the SCA7 Purkinje neurons, as you can see here. But this is rescued nicely back to normal levels by SIRT1 overexpression. So you have a rescue that's of phenotypes of a histological nature, as well as a behavioral nature, transcriptional nature, and also electrophysiological. Okay, so then what about NAD? Well, we wondered, is it possible to rescue phenotypes in the SCA7 mice simply by elevating the levels of NAD? Now, NAD is a really important cofactor. We think about it a lot in terms of metabolism. And I think evolutionarily, that sort of is its sort of like a moonlighting job, because originally its main role was to serve as a cofactor, for important enzymes. And NAD, the way we make NAD is most of our NAD is made through the salvage pathway. We don't synthesize it de novo. So when we use NAD, we basically end up bringing it back through this pathway, and then we have these enzymes that drive its production from MNN to NAD, and there's 3 of them, and they're in different subcellular localizations of the nucleus, cytosol, and mitochondria. Anyway, there's lots of different ways you can imagine for increasing, where you can come in with different NAM. But what we did is we were interested in nicotinamide riboside as a way which gets phosphorylated to NMN and is a way to prime this sort of salvage pathway. So we decided we would supplement the diet of our SCA7 mice with NAD, and Paul Swiatkowski, my Polish postdoc, did this work, and we were able to improve behavioral phenotypes and also to extend survival significantly, just based simply upon NAD repletion. And we also looked at the neurons derived from SCA7 patient stem cells, and there, if we supplement it with NR, we could rescue problems with calcium influx and also with calcium flux in terms of you can see the variance is quite abnormally high, and that can be rescued by NAD. And of course, NAD is being utilized by PARP. And so we went we got postmortem material from SCA7 patients and from matched controls, and we immunostained for PAR, poly-ADP ribose, the product of PARP1. And you can see in SCA7 that it's increased. We quantified it two different ways. We determined the percent of cerebellar neuronal nuclei that were PAR positive, and that was significantly increased. Or we just measured overall PAR immunoreactivity, and we saw that was significantly increased, suggesting that hyperactivation of PARP1 could be important for SCA7. And so, what comes out of this work is this pathway where we think that there's DNA damage leading to PARP hyperactivation. You get NAD depletion, and that leads to an inhibition of SIRT1 enzymatic activity. So the client proteins of SIRT1, including importantly PGC1 alpha, are impaired, and so then you have a problem with calcium homeostasis, inositol triphosphate signaling, and you get neurodegeneration. And so in terms of selectivity, which I mentioned at the outset, which I'll tell you about, first I want to talk about the therapies. So one way to treat this possibly then would be to use PARP inhibitors, of course NAD repletion, SIRT2 activating compounds, which are elusive. Resveratrol has been suggested, but that's very controversial. We're thinking about PPAR agonists. And you could also use modulators of potassium channels that regulate calcium channels. And this SK channel modulators, positive allosteric modulators, actually are moving forward into the clinic as we speak. But yeah, the idea I wanted to mention is that I now think that cerebellar degeneration, there's a catastrophic cliff that starts with DNA damage. And indeed, there's much genetic evidence to support this. There's many DNA repair genes that if you mutate them in the human, you get a cerebellar degeneration. Sometimes you also get cancer predisposition with that, but sometimes you just get the cerebellar degeneration alone. We think that leads to a mitochondrial or metabolic stress, and ultimately you end up with calcium dysregulation. That's the final sort of step in the sort of decompensation of cerebellar function that leads to clinical symptomatology. Okay, so I'm going to now transition. Yes, and if swimming with whale sharks or meeting bears in the country are too dangerous for you, you can always visit a koi pond, as an alternative outdoor activity. Okay, so now I'm going to talk about therapeutic strategy. So what's the best potential therapeutic strategy for SCA7? Well, the primary cause is production of the mutant protein. That's what leads to the retinal neuron degeneration. So if we prevent the mutant protein from being made, we should prevent the disease from happening. It's that elementary, right? So before I even moved to UCSD, Ionis Pharmaceuticals had contacted me for a potential collaboration, and I was thrilled about that. And so Ionis Pharmaceuticals is in the business of making oligonucleotides, and for gain-of-function disorders, the idea is to make antisense oligonucleotides, or ASOs. And the strategy here is to target the RNA that encodes the disease protein. So what is done is you make a 16 to 18 length oligonucleotide with perfect complementarity to the target RNA. It has lots of modifications, but it still has to be enough like DNA, so you form a heteroduplex. Because when you get the heteroduplex, that's a substrate for degradation by RNAH. And you get degradation, of the RNA, whereas the antisense oligonucleotide survives and goes off to bind to another RNA. And so that's what gives you the longevity of the effect. And oh, this work, by the way, has been led by Chen Liu, who's a project scientist here in my lab at UCI. But she started this work at UCSD and finished it at Duke, actually. Okay, so our approach for delivery of the ASO is to direct into the vitreous humor. So, you know, for this audience, I think everyone knows the anatomy and what we're talking about here. And so we did some experiments and to see if we were successful. So in the right eye, we're injecting ASO, in the left eye, just PDS. We have an antibody that detects the ASO backbone, and you can see that the ASO, you know, nicely goes throughout various layers of the neural retina. So the intravitreal delivery method is quite effective in mice and should be in humans. So then what we did is we tested different ASOs, and we saw a nice dose response for one particular ASO, where as we injected more, we saw a reduction in the levels of the ataxin-7 mRNA. And then we figured we should proceed to do a preclinical trial. And this first trial that we did is a so-called prevention trial. And we're using these knock-in mice. Now, these mice develop symptoms around 6 to 7 weeks of age in terms of vision, and they go blind by about 11, 12 weeks of age. And so time zero is for us 4 weeks of age, where we would do our injection of ASO, control ASO or PDS in the left eye, and then our experimental ASO on the right. We do baseline measurements of ERG, make sure there's no outliers or anything weird going on, and make sure and establish our cohorts. And then after 4 weeks, we come back and we do another set of ERGs. We take a few mice for histopathology, but then we keep a number alive and come to a terminal, you know, time point at 10 or 11 weeks of age. And so that's how we did this preclinical trial. And this work was funded by the Gunn-Harrington Award that I was fortunate enough to receive. And so what we found is if we took our SCA7 mice and we did IVI injections of ataxin-7 ASO in the right eye, we saw a significant reduction in the formation of aggregates, as you can see here. And here we did filter traps and quantified it. And so you have a reduction of about 60% in the accumulation of the misfolded protein. And in terms of visual function, here with the ERGs, if you look at the cone response, in the treated mice, you have a good retention of function, much greater than in the controls. And you know, as the disease progresses, the cone function, you can't get it back to normal levels. This mouse model is just too aggressive for that, but you do show a significant retention compared to your controls. Where rod function, it's a little bit different. You can see retention of normal function at the initial post-intervention time point. However, as the mice are reaching the terminal stage, again, you have nice retention compared to controls. You know, so again, a really robust therapeutic response, but not back to the, it doesn't stay at the level of normals. So after we did this work, oh, one other thing I wanted to show you is you know, this epigenetic dysregulation that ties into the earlier discussion we had of CRX and STAGA. Of course, we looked at histopathology, and we saw, you know, that there was rescue in terms of the thickness of the ONL. We also looked at the expression of cone-expressed genes in terms of if we could rescue, you know, those as well. And again, you know, it was okay, but not awesome. And then we also looked at this H3 ubiquitination mark, where you see a problem with deubiquitination. So you see increased ubiquitination in the SCA7 mice. And we looked at histone H3 acetylation. And again, we saw, you know, rescue of these chromatin remodeling activities in the mice, in the eye, the right eye that was treated with the ASO. Now, so we did the prevention trial, and then we said, okay, let's do an intervention trial. We developed a less severe SCA7 mouse model that had 210 repeats instead of 266. So, you know, the progression plays out over a longer period of time. So here, the time points are slightly different. You start at 9 weeks of age. You wait till they're about 15 weeks of age to get your first set of ERG readouts. Then you wait another 3 weeks to 18 weeks of age, and then that's the terminal endpoint for the study. And so here, what you see is that you see improvement in terms of a reduction of the accumulation of the misfolded protein in terms of cone function. Again, you see that the treated mice do better than the mice that are treated with control. Again, it's something for the cone function, doesn't blow your mind. The rod function that you retain, much more impressive. But again, these mice also, have a very short lifespan. They only live twice as long as the other mice, so they die around 22 weeks of age. So again, it's in a human, this would be like an infantile onset situation. So it's a very dramatic phenotype, and we're pushing against a very heavy weight here, and we're managing to budget. So I interpret this very positively in terms of the potential of this therapy for being meaningful in human patients. And so how close are we to doing this in humans? Well, we're getting there. So we put together this data package, and our colleagues at Ionis, who we worked with, presented it to their scientific board and to Stanley Crook back at the beginning of 2019, and it was approved that they should go forward and they voted in what they call target sanction. They voted yes for target sanction. And now, so at that time, the production of human ASOs, was begun, because, in this study that I showed you, we're working with ASOs that are directed against the mouse gene, so they're not complementary to the human. Anticipation of this work, we had to create back transgenic mice that express the full-length human ataxin-7 gene, because that's important for the purpose of ASO screening and selection. They need an in vivo system, but they need it to be the human gene because they need to generate human ASOs. So that's work that we did, and we generated those mice and provided them to it.
Dr. Rani, we have this video we've been showing.
And hopefully, if things proceed as planned, we'll have a lead ASO soon, and we'll be able to apply for an IND and start a clinical trial. Now, in terms of the clinical trial, so we're very fortunate. So I was an MD PhD student at the University of Pennsylvania, and Brian Brooks followed me in the same lab just a few years after I finished. And so we've been friends since then, and he is now the clinical director at NEI. And about, I guess, 8 years ago, I approached him and asked him if he'd be interested in working with us on SCA7, and he very kindly and enthusiastically agreed. And that led ultimately to this clinical natural history study, which, as I said, began about 5 years ago. And it's led by the NEI, but it also involves NANDS. And Brian is the PI, and Larissa Huron is the co-PI, which means she does all the work. So the purpose is to identify ophthalmic outcome variables in anticipation of a clinical trial, and to understand the ocular and neurological natural history of SCA7. And the plan was to recruit at least 24 patients and follow them annually over 3 to 5 years. So it only took us about 1.5 years to complete the enrollment, which was about 2.5 years ago. And a number of patients have returned for follow-up evaluations. We have a lot of longitudinal data. We're getting a good sense of metrics for the clinical trial. Though my understanding is the FDA is really only interested in visual function, so all these cool tests that we are identifying, FDA probably won't be as interested in them. But anyway, that work is going on, and it's been a wonderful collaboration. I'm a co-PI, on this natural history study as well. And NEI is committed, or at least their former director, Paul Sieving, was committed to supporting a clinical trial of SCA7 to treat the retinal degeneration in the near future. Okay, so with that, I'm going to stop. So this is sort of what the group looked like, and now we've been reconstituted. This is the current group. It's quite robust. I was fortunate in that 13 members of my lab moved with me from Duke here to UCI. I've mentioned some people along the way. I've mentioned a number of collaborators. I think that Chris will remember these folks from our time together in Seattle, hopefully fondly. My Ionis partners have been really great. But I really also want to point out, this just isn't possible without the patients. And the patients have been great, and the families. This is a really bad disease. So I often wonder why they're so great, but they are. So hopefully we can help them soon. So with that, I think I'll stop sharing my screen. And if people have questions, they can.
Thank you so much. Let's move to questions quickly. We have a question from Dr. Brown.
Hi, thank you for such an interesting talk. Early in your presentation, you showed that CRX is dysregulated in its transcription. And I was wondering in that panel of, I think, 92 genes, where you looked at SIRT1 rescue, if that was one of the genes that was rescued, possibly. And then the second thing is, showing that there's increased reduction in NAD in cells that are affected, kind of suggests that there might be a way to image that in vivo using Gragene's microscope, where you can look for changes in metabolism in the photoreceptor layer, and even look for that increased oxidative stress, like Enrico Grattani has shown with long lifetime species causing oxidation of membrane lipids and maybe even photoreceptor disc lipids. So two kind of separate questions, but I think it's really interesting to see all these connections that you've shown.
I'll start with the second question first. I mean, the experiments you propose sound really exciting, and I'd be very interested in learning more about them and how we could do such experiments. I'm very interested in dynamic visualization of NAD, and the retina is a wonderful place to test these hypotheses, and we haven't really evaluated this thoroughly yet in the retina. That was waiting for a future day. So maybe that day can come very soon. That would be wonderful. In terms of CRX, so CRX expression is restricted just to the retina and to the pineal gland. So yeah, we wouldn't expect to see CRX target genes or CRX itself as relevant in the cerebellum.
All right, let's move to Corinne.
Yes, hi, thank you. It's a very exciting and very interesting presentation. Quick question in terms of retinoids. You mentioned their involvement in the pathway. Have you tried in your mice any agonists or antagonists to test that hypothesis?
I'm sorry, to test the hypothesis for which?
If they did anything, the agonist or retinoid agonists or antagonists.
Oh, so retinoid agonists or antagonists?
I mean, PARP.
Yeah, right. PPR. Yeah, so have we tried, right, so PPAR delta is our favorite.
Yeah.
There's other options, as you point out. No, we haven't done that yet. Again, that's something that we're interested in doing. And actually, I'm very interested in, advancing further studies of nuclear receptors in retinal disease. That's an area that I was thinking of getting into, something I wanted to discuss once we're completely settled in, or maybe we'll never be completely settled in, but settled in enough in the new year. With the vision research community here, I have some ideas of experiments that I thought it might be fun to do, experiments that might bear relevance to other retinal degenerative disorders like AMD, but we haven't done those experiments yet.
Thank you.
Hi, Al. This was wonderful, and I really appreciated your update and some wrap up of the older studies. So first of all, I am ready for discussion about what you just mentioned. This is very interesting, and as you know, I'm long involved in the transcription regulation. But one of the things which I don't know, maybe I missed, but what is actual role of Ataxin-7? I mean, is there any enzymatic role? Is there any activity? And what signaling pathways make it to shuffle? I still wonder.
So you're asking what the function of Ataxin-7 is?
Yeah.
Yeah. So we think that Ataxin-7, basically it goes into the complex and it helps to organize the physical interactions of other proteins in the complex. So it's just like an adapter protein. Why, but it's really, why it shuttles in and out of the nucleus is a mystery. And we don't have an answer for that. And the complex is very much tied up, has a dedicated subunit for shuttling mRNAs in and out of the nucleus in yeast, but that subunit isn't conserved in humans or mammals. So the hypothesis we tested, but we really couldn't demonstrate that to our satisfaction. But I've wondered a little bit about that we should look at that more carefully. Yeah, but what's clear is that if you knock out ataxin-7 in mice, that it appears to be pretty well tolerated. Maybe a little problems with learning in the hippocampus emerge. It turns out ataxin-7 has 3 other proteins in its family: ataxin-7 L1, L2, and L3. And ataxin-7 L3 is in the complex and has a close relationship with USP22 that mediates the histone deubiquitination. And it appears that maybe it can substitute for ataxin. It can compensate, when you have a knockout situation. I mean, that's why we think if we knock it down, because the ASO therapy, I didn't mention this, is it's not allele specific, so it's allele non-specific. So you're going to knock down the normal, RNA as well as the mutant RNA, and some people might worry about that. We don't think there's any data. In fact, we've done experiments and we don't see any evidence that when you knock down the normal, and at least in the mouse, that it has any deleterious impact on visual function. So ataxin-7 is a transcription factor that is part of a complex, but there's a lot about its normal function that we still really don't understand.
All right, let's move to Chris Kenny. We have still a few people who wanted to ask.
Hi, that was a really amazing talk. That was really terrific. I'm very interested in the mitochondria aspects, and I have basically 2 questions. When we have a model where we have cells, they're cybrids, where all the nuclei are the same, and then we can take the mitochondria and get them from people from different maternal backgrounds. So maternal African, maternal European, or from diseases, AMD versus age-matched normal. When we do that, we find that the mitochondria, through retrograde signaling, influences the epigenetics. It will change the expression patterns for HAT and some of the deacetylation genes for the methylation genes. So my question would be, have you looked at the mitochondria or the mitochondrial genomes, either in the patients or in your animal models?
No, we haven't done that. So it sounds like you're a disciple of Doug Wallace.
I am. He introduced me to mitochondria, yes.
Doug's a good friend. So yeah, I think that these are really interesting ideas. Yeah, I think it's also interesting for SCA7 because half of our patients are of an African racial ancestry and half are Caucasian, well, roughly.
Programs may
Caucasian European. And there's some differences in terms of anticipation and phenotypes. So yeah, the mitochondrial connection is something we haven't thought much about.
So if you have DNA, you can easily look at the haplogroups, and then we can also sequence and see if there are differences, because we find that with certain diseases, I'm going to make it really short, with Africans with diabetes, when those hybrids, it changes the methylation pattern and they become resistant to demethylation in the cybrid cultures. So there's a lot of influence that the mitochondria and the mitochondrial genome and the haplogroups have on the disease, on disease pathways. So maybe
something I'm definitely interested in this. We're actually about to, we were launching a huge ascertainment of patients before the pandemic hit. So we weren't able, I didn't want my SCA7 patients going to the clinic to get their blood drawn and things like that. So we had to put that on hold. But this is a line of investigation that we're pursuing, and the types of experiments you're describing. I think, yeah, I'd love to talk about talk with you more about it at some point.
Okay, I'd love that.
All right,
thank you.
UWMC.
The question I have is related to the vulnerability of protein folding comparing cones and rods. I assume this mutated gene is expressing both in cones and rods, and why the cones are more vulnerable to protein aggregation, but not targeting rods.
Well, I've always sort of thought that the cones were more vulnerable simply because CRX, was, one of the top clients of the Spada complex, and CRX's impact, plays out more prominently in cones rather than rods, which explains when you have genetic mutations in CRX why you get a cone rod dystrophy instead of, like RP or whatever. So I've been looking at it from a more of a genetic perspective as opposed to a biochemical explanation. But the underlying question of whether rods and cones deal with misfolded protein stress differently, I don't know the answer to that question. I don't know if anyone's ever even looked at that. So it's an interesting question in its own right. Of course, misfolded proteins in RP occur, but I think they're restricted to rods. So I don't think there's anyway, yeah. So it's, I think I think there's an interesting idea there, but for SCA7, I think we have an explanation that's more, based upon molecular genetic data that to me, I think seems reasonable.
Okay, we have two last questions, and Henry, are you there? Clausen? Okay, so let's move to
hard to unmute.
Okay, good. So you will have a final comments if you don't mind, but we moved before.
I thought this was really fantastic. We had an operational emergency on the East Coast, so I didn't catch as much as I wanted, but there was enough there to get me madly scribbling notes that I'd love to follow up on. So really fascinating stuff.
Okay, the last two questions will be Dr. Gong and Emma.
Hey, Alberta, truly enjoy your talk. If you go down to Baja next time, please be taking me around to see the shots.
Sure.
So I have a very general question with the origin of the aggregation also of this polyglutamine protein complexes.
Okay.
So does this aggregation require the second modification of these proteins, or what is the general knowledge about that? I mean, the second translational modification with the proteins.
Yeah. Well, I mean,
which to do,
right.
Yeah, which maybe to do with the vulnerability of the cell types.
Well, I mean, I think that there are post-translational modifications are important for the disease process for various reasons, it would appear. And that relates to, pathways in terms of subcellular localization and all sorts of other things. But I think that the basic concept is it's just simply the length of the glutamine track. Once you reach a certain length and you exceed it, then this misfolding event occurs where you get this like, this sort of beta, sort of the polar, beta formation where it sort of goes back on itself that, as I said, Max Perutz described way back, in the mid 90s and wrote a book about it. So yeah, there are certainly other, aspects probably that are modifying, the polyglutamine misfolding, but it appears that length is king. And the longer the repeat is, then the more aggregation and misfolding you get. So it's a function of size. Undoubtedly, there's other factors, but in this case, it appears that size matters the most.
So you prefer the vulnerability will come out of the targets, basically the aggregation happening in all cell types. However, the target is where the cell vulnerability is.
Yeah, well, so that's actually another important point. So the way the cell deals with the misfolded protein, well, different cells deal with it differently.
Yeah.
And the ability to refold the misfolded protein, some cells have more capacity at that. The ability to sequester the misfolded protein into, aggregates that basically where it's neutralized and not causing a problem, that varies from cell type to cell type. And then, the interactions in the soluble phase, we think drives the disease process. So the misfolded soluble protein, what it's partnering with in different cell types, we think is what drives the selective vulnerability. So anyway, that's sort of those are some of the overarching models, for the field, if that helps.
Thank you.
Sure.
Emma?
Hi, Dr. Spada. My name is Emma. I'm Dr. Current postdoc fellow, and I have 3 questions though.
They have to be short,
Yes. Yeah, the first one is the phenotype of this ataxin-7 resembles to me ciliopathy. So, and you said that the protein translocates from the nucleus to the outside. Have you looked if it forms part of the cilia structure?
No, we haven't looked at that.
You haven't looked at that. The second question that I have is about your oligonucleotides. I know that this type of therapy causes off-target effects. Have you seen those off-target effects on the retina? And if you don't, how do you escape from that?
I don't, so the oligos are directed against the ataxin-7 gene with exquisite, selectivity. So, there's.
Yes, but that is very hard to achieve.
But not, yeah, but I mean, the whole, what Ionis does is they do a lot of screening and they try and, rule out the likelihood of off targets. You may get some, but, again, that's part of the whole sort of pipeline that they're devoted to. It's a concern, but it has not prevented the deployment of these biologicals as an effective therapeutic. And we have many that are moving toward, I'd say, clinical approval certainly for ALS type 1 against SOD1. And we're in phase 3 for Huntington's disease. That looks really good, the data, but it's still too preliminary to call. So yeah, this is a concern, but it's a tractable concern. Now, other types of oligos that are, like directed against the CAG tract or whatever, that would be a real problem for off target. Yeah.
Yeah. And the last question that I have is, have you considered about a therapy of clipping the poly-Q and then, just reduce the elongation of that part of the protein and just.
You mean like a genome editing type of approach?
Kind of a editing type approach, yes.
Well, of course, that's the dream, to have a one and done approach. But, the challenge is transducing, the cells throughout, post-mitotic cells in the central nervous system and blah, 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 blah. So it's, and we also think, Burton and glia are involved, so astrocytes and yeah. So yeah, I mean, we think about that and people write grants and get their grants funded, but if I'm a patient or a family member of a patient, I'm thinking like, that's still, far down the road in terms of its potential realization.
Also, following on Emma's question, your oligo is against mouse protein or you humanize those mice? You didn't mention that.
No, this was against mouse.
So the same work will have to be done on the human. Are you planning to humanize this in mice?
Yeah, that's what we did. Well, at the very end, I mentioned we made back mice that expressed the entire human ataxin-7 gene. And that was a prerequisite for Ionis to move forward. And so we made those mice and then we supplied them to Ionis and yeah, they're being used for screening.
But with different set of oligos, right?
Different oligos, absolutely. It's a whole different pipeline. They did the mouse pipeline for us for the proof of concept work. And then when that work, was satisfactory, they started the human pipeline. And so that's sort of, and that was started now 2 years ago. So hopefully, we'll have something soon that, we'll go into safety testing in rodents and into non-human primates.
I promise you, this will be last question. On those sections with oligo, you have a lot of dots, fluorescent dots throughout this section. Is that an artifact or there's something that relates to the oligo? If you look on the slide, it's clearly a lot of fluorescent dots throughout.
Yeah, I think that has to do with the accumulation of the oligo in certain regions where, at least I always interpret that as real and not as an artifact. Okay.
Okay, thank you so much. This was truly wonderful. and good to have you here so we can torture you more. And
thank you. It was really great.
Yeah, well, I can't wait to interact with you guys more, and thank you very much, everyone, for attending. And everyone, if I don't see you, have a great holiday, and hopefully